I'm your host, Nick Jackomis, and today I'm speaking with Sean B. Carroll. Sean is an evolutionary developmental biologist. His lab studies how novelty evolves, how new structures, new forms evolve in the animal kingdom, and what the molecular genetic basis of those things are. So he's both an evolutionary biologist, someone who studies how organisms change over time, and also a developmental biologist, someone who studies how animal bodies are put together. And so he's really good at thinking about how tweaking or changing the process by which animals are constructed over the course of development results in evolutionary change over time. Sean is the author of many popular science books about evolution, including Endless Forms Most Beautiful, which is probably one of my favorites. It's actually a book that made me want to study evolution. I actually worked in Sean's lab when I was in college for almost 5 years and spent a lot of time thinking about some of this stuff. Sean went on to do a number of other things. He's had a lab for many years now, but he was also the VP of Education for the Howard Hughes Medical Institute and oversaw one of their production studios. So he was involved in making a lot of educational content, science documentaries and things like this. And he recently stepped away to go back to the lab full-time at the University of Maryland. And so I talked to Sean about all sorts of topics to do with evolution and development. How are animal bodies put together? What are the molecular genetic mechanisms that govern some of that? How does evolution work? What is natural selection? What is sexual selection? How do mutations accumulate? What kinds of mutations happen in what kinds of genes to create certain types of phenotypic or anatomical change? We talked a little bit about the evolution of snake venom, which is what he's studying now in the lab. We talked about human evolution. We talked about the speed of evolution. So if you're interested in animal diversity, how animals are put together and how they change over time, this is a really interesting episode. Sean's a phenomenal science communicator. I had a lot of fun catching up with him on the podcast. As always, I'll remind you that I have a Substack, mindandmatter.substack.com. You can subscribe to my free weekly newsletter or support the podcast by becoming a paid subscriber to get early access to episodes and other paid subscriber benefits. I put all of my podcast content on that Substack, as well as my long-form science writing, which synthesizes and integrates a lot of the topics I talk about across different episodes. So please go check that out if you haven't already. And with that, this episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Here's my conversation with Sean Carroll.
I was doing double duty there as both VP of Science Education and head of the studio. 13 years as VP, 6 years as head of studio. That was incredibly busy, and you cannot sustain that for infinity.
Yeah.
So, I thought it was time to step back, and I had my lab here. I was going every Friday to the lab, but I thought I kind of feel like I'm refilling, the well now, back in science for a few months, enjoying reading, enjoying daily conversations about research in the lab. Still have a hand in the film storytelling, but I'm not responsible for the production, the enterprise. So yeah. I am in a good place.
Okay, great. What? How big is the lab now?
8 people.
Oh, that's a good size, I think.
Yeah.
Yeah.
It's enough for things to be happening and not so many that I'm overwhelmed and, you know, stretched too thin. So
Yeah. What well, just for background, for people who don't know you, what do you study generally? And then can you just give a snapshot of some of the projects you're working on right now?
yeah, the central question that's guided things for a long time is the origin of novelty, where new things come from in the course of evolution. And that's really things that are sort of qualitatively novel, that either it's body parts that do something new or molecules that do something new or and give some sort of capability to an organism it didn't have before. And I've decided to focus a lot of energy on the origin of snake venoms and their toxins. Because the fundamental reason why that's interesting, venoms have been invented multiple times in the animal kingdom. You know, spiders and scorpions and octopi and snakes and all that kind of stuff. We've come up with venoms independently many times. And the question is, where do those things come from? Are those sort of normal body proteins that you've sort of hijacked to do something new, or you really have, has evolution really stitched something together that didn't exist before? And in snakes in particular, there's lots of just the history of snakes, which is underappreciated. How they've sort of invaded continents relatively recently in evolutionary terms and flourished, radiated into all sorts of species. They're really underappreciated as models of evolution.
And one of the things I want to talk about first with you is, you study evolution. Obviously, we're going to talk about a lot of evolution stuff. I can remember being 17, a high school student, and I knew what evolution was in the abstract, at the level that you learn about in high school biology. I knew at the time, millions and millions of years go by, the DNA mutates, new things emerge, but it was all kind of abstract. And it wasn't until I read your book, Endless Forms Most Beautiful, that things kind of clicked and became much more concrete and just easy to intuit. And I think the reason for that is just, if you are trying to understand evolution, it makes it a lot easier if you first understand something about how bodies are built in development. And then you realize, oh, if I just sort of tweak this process, it just makes everything a lot more grokkable, I think, to the mind.
Yeah. So we can all appreciate, we all developed from an egg, right? A single-celled egg made all these body parts. So, changes in anatomy are due to changes in development. And you can appreciate with all these different processes going on that, a little tweak here, a little tweak there can make a pretty big difference. And, we had no access to understanding those tweaks, really until the 1980s. So that this interested biologists for a really long time, but we couldn't make it concrete until the 80s and 90s, and really get into the actual mechanism of how bodies are built and how they change. Mm-hmm. And that I think that changed a lot of things for the way we could talk about evolution. Evolution has this, evolutionary science has a big theoretical history. There was a concrete history that Darwin gave us, but there was a big theoretical period of, okay, how do small changes add up to be big things? But we just couldn't get to the concrete. And I think a lot of people, including me, need the concrete. I want to look at creatures. I want to look at parts of those creatures. I want to know why those parts matter in their, in the world they live in. And I want to know how those parts change. And then now it's like I got a full description, of the, owner's manual of how things work.
Yeah. And I think part of what you're referring to there is, there's literally, between Darwin and Darwin and, the 1980s developmental biology, there were decades and decades of research happening, but a lot of that was basically applied statistics, and it was the math to figure out, and the number crunching to literally calculate, like, okay, if you've got this much time and this much DNA, yes, you can account for things, but it was literally like math, and it wasn't like mechanistic biology.
Yeah, it was mathematical. It was heavily mathematical models. Brilliant people. I mean, especially in the 20s and 30s. But realize if a lot of that math came before we had the double helix of DNA. Mm-hmm. So, it's not till 1953 that really the world sort of understands that what the genetic material is. There was scientific evidence that it was DNA from some years earlier, but that was not kind of widely understood. And it was really the structural model of DNA that we said, okay, here's the molecule that transmit the information generation to generation. And it was instantly obvious to Watson and Crick how mutation happened. It was a change in the sequence of bases, and that would change the characteristics of organisms. Now we have concrete for the genetic material, and we know that evolutionary change must be due to changes in genetic material. So we really just couldn't put, that kind of foundation underneath Darwin until we were able to look at the stuff that's changing in evolution.
Mm-hmm. And, one of the things I want to ask you about here, when I first started learning some of these key facts, I remember being blown away. And the question I'm going to ask you is, how many genes does a human being have? And I want you to put that in historical context for us by telling us how many genes do we know we have today, and what was the thinking before we actually did the Human Genome Project and actually could look and see?
Yeah. So the question of how many genes any creature has, that was a question I got a sort of a front row seat to, because the technology for sequencing DNA sort of sprang up just as I was hitting graduate school. That was then starting to be applied on a larger and larger scale, but there was still a good decade and a half between sort of being able to sequence a gene and being able to sequence a genome, the entire genetic complement of an organism. And I would say for most of that time, well, there are several. I'm going to start with this as sort of like several biases that I can recall because I was exposed to all of them. Biases in human medicine, biases in biology, but a huge human bias about human biology that somehow we must be the most complicated, right? Now, you would be better situated than I am to comment on that from a neurobiological point of view. But from a genetic point of view, there was an anticipation that, we would be, genetically more complex than any creature on the planet. Well, it took a while to get to that answer, but, the first picture we got was probably of a bacteria. And those are sort of on the scale of 4,000 or 5,000 genes, essentially, run the metabolism and physiology in bacterial cells. Mm-hmm. And then it took a while to get to animals like the fruit fly Drosophila or the nematode worm Cenorhabditis. And in those cases, we were probably getting into the ballpark 13,000, 14,000 genes, something like that. And then the question would be, what would humans have? And look, one of my heroes, Jacques Monod, I think I have him on tape somewhere. There's a video recording him somewhere. This is in the 60s. Just extrapolating, because we knew that of the size of the amount of DNA in a human cell, that humans might have 2 million genes. Mm-hmm. And for a while there, I think the number that I heard circulating might be like 100,000 genes. So still, far more than other animals or whatever. And I remember having this discussion with a really prominent fruit fly researcher who had a good grip on things. And he said, well, Sean, what do you think? And I said, it's not going to be much more than a fly or a worm, 15, 20,000. Bingo. Maybe it's 20,000, but it's not any more than a mouse or something like that. Mm-hmm. So, first of all, that kind of, vanquishes our genetic, specialty, our specialness genetically. But it really underscores something that, as you know, is fundamentally interesting to me about evolution, which is how you build diversity with essentially the same toolkit of genes. Mm-hmm. So you don't need more genes to make more and different kinds of creatures. You can use the same genes you got, just in myriad different ways. So in many ways, sort of the leveling of the playing field for humans to have a similar genetic complement as mice and, chickens and even fruit flies was to also tell us that we shouldn't be thinking that the number of genes dictates the complexity of a creature, but it's how genes get used that are the real source of diversity. And that is a major message that then emerged in research that was directly comparing how different animals were built and what the genetic machinery involved was.
Mm-hmm. Yeah. So just the simple observation that we don't have that many more total genes than a fly, and we've got comparable numbers to, and sometimes even less than, creatures that we would basically universally agree are simpler than us. That tells you that, it's about how you use this stuff much more than it is the amount that you have.
Right.
And obviously you're going to tell us a lot about the how you use it part of it. I want to build up a little bit of vocabulary for people. So when we talk about genes, what exactly are we talking about in the DNA in terms of like protein coding sequences versus other stuff?
Yeah. So let's do a little accounting. So basically we've got 23 pairs of chromosomes. Okay. A chromosome is a long molecule of DNA. A gene is a segment of that molecule of DNA that has some information that gets used in cells. So, if you might say maybe on average, there might be 1,000 genes per human chromosome. I'm averaging things out a little bit. So an individual gene is going to take up a certain amount of linear space on a molecule of DNA. That gene, a segment of DNA, in order for it to then contribute to physiology or development of an organism, is going to have to be used to make as instructions to make proteins. And there's an intermediate in that, which is it's first transcribed into RNA. All this language, all this cryptographic language, by the way, came from the discoverers who all had kind of had a World War II background, right? So the gene is transcribed, right? So you make this intermediate transcript, and that transcript is translated, decoded into making protein using genetic code, sort of like, a code book or, think of things like the Rosetta Stone.
I never actually thought about that the discoverers here were sort of operating in the Oppenheimer era.
Yeah, they're coming out well, and Crick, who was was definitely the most brilliant theoretician in biology. Crick worked for the Admiralty in World War II. So all these folks, a lot of these folks had, if not a direct World War II background, they were of the era, just coming right out of World War II. So they had this, code books and transcripts and translations and decoding and deciphering. All that language was there. But anyway. The point is that when we think about a gene, we're really talking about a stretch of DNA, a linear sequence of DNA, 4 letters, might take up thousands and thousands of letters, maybe even 100,000 letters for a gene, but still it's a discrete segment of DNA. And that information is in a stepwise process transcribed and translated into the making of proteins. And proteins are linear chains of molecules called amino acids, but those proteins do essentially all the work in our bodies. They're the things that carry oxygen. They're the things that fight off invaders. They're the things that digest our food, et cetera. And really, to operate a cell, just to do the work of any old cell, looks like you probably need 5,000 or 6,000 genes working. So some of the genes we have are specialized to various cells. We have genes that, as I said, that carry oxygen. They're in our red blood cells. Genes, they're active in our red blood cells. Genes that fight off invaders are active in our white blood cells. Genes that transmit electrical signals, those are in our neurons. So, genes that build bone, and proteins that build bones, those are, of course, going to be in cartilage and bone. So, we've got a lot of things that are specialized, but we also have a core that we share with almost everything on the planet to just do basic metabolism of a cell.
Yeah. So things like, literally making ATP, making energy, or how the basics, the fundamentals of how a cell divides, things that every single cellular creature has to do.
Absolutely. And this stuff is deep. So we can see that stuff is shared way back. That's shared throughout. And there's many things we share with bacteria. There's lots of things we share with simple unicellular creatures. And so we know that stuff goes back. In fact, the genetic code we use is exactly the same genetic code that a fly uses, that a bacterium uses, et cetera. So the genetic code is universal, or universal with respect to planet Earth. But that tells you that stuff is old. But that machinery for doing all that stuff has been around a very long time, and every cell needs it.
And when we say, we often talk about genes with respect to proteins, like a gene encodes a protein. Right. If you look at the whole genome, roughly speaking, what percentage of the letters are the code to make proteins versus other?
I think in the human, I'm not going to give you a precise figure because it's kind of left my aging brain. It's probably on the order of like 2%.
Okay, so but quite a small amount. It's not
quite a small amount. So there's tons of stuff, tons of other DNA in our cells not devoted to encoding proteins.
Mm-hmm. And the term, I used to hear this term a lot more. I don't hear it as much today. I've also probably tuned it out because I do know it still gets used, is junk DNA. And I think the idea historically, you can correct me if I'm wrong on the details, is, the Human Genome Project came out. We started looking at the actual code more. We realized, okay, we only have X number of genes. We only have, 2%, give or take, of the genome devoted to encoding proteins. And so there's all this other junk DNA.
Yeah.
What is junk DNA? And is that term even make sense today?
No, it's a bad term because it lumps, and we don't want to lump. We don't want to lump. It was appreciated, and this is it's kind of an inconvenience to biologists, and it's kind of aesthetically not pleasing. But there's reasons why genomes can accumulate a pretty large amount of like repetitive DNA. So we get exposed to agents like some viruses that insert DNA into our genomes. There's processes going on where DNA can get amplified. And that doesn't mean that DNA necessarily contributes to how our cells perform or anything that we do. So humans, I'll just use humans as an example. There's not necessarily a lot of pressure to kind of prune out DNA. So you can sort of think of it somewhere it's like, kind of like the garage or some storage shed you've got. Stuff can pile up there whether you're using it or not. Over evolutionary time, it can pile up. But in that garage or in that storage shed, there are some useful tools, and there's still a small percentage. So it's better to sort of talk about the coding fraction, which is small. There's a non-coding fraction, which is very large, but some small part of that coding fraction is also crucial machinery that governs how genes are used. And I think the best category to sort of describe these are things like genetic switches. Mm-hmm. These are sequences in the DNA that the way they operate in the process of turning genes on and off. And so it's not all junk, right? The non-coding DNA is not all junk. There's a part of that non-coding DNA that it's crucial to the whole choreography of which genes get turned on and off in your cells, and in which cells, and at what times in the development of an individual. That stuff was really hard to find. Okay, so I can give you sort of a historical reason why junk has, kind of held on. First, people found, and then in some other species like some amphibians, there's a massive amount of DNA that doesn't encode anything, and people are like, junk. Yeah. But because there's a universal genetic code, it's very easy for our computer programs to identify stretches of DNA that encode proteins. Mm-hmm. Bang, easy as can be. It's not easy to identify among that other DNA which stuff operates in some important function and which stuff is just going along for the ride, which stuff is just that, that stuff in the garage or the storage shed.
And it is some combination of both. Is that true?
Yeah. Oh, for sure. Yeah. So that has to be figured out experimentally. There's no kind of computer program that will tell you this segment of DNA exactly operates this way. Mm-hmm. So it was longer to realize that probably in the human, I'm going to say about 3% of the DNA is devoted to orchestrating how that 2% of the DNA gets used. And all that stuff is really important. That's what orchestrates, that's how you put a head in the right place and make the right number of red blood cells and, lots of other choreography of how genes get used.
Mm-hmm. One, I want to give people a sense that there's different types of genes encoding different types of proteins. And so one distinction that I think is useful is, there's a lot of genes that encode proteins that do like the everyday housekeeping physiology stuff of the cell. So enzymes that chop up certain, nutrients or, carry oxygen around or whatever. They're doing the daily operations of the cell.
Right.
And then there's other proteins. And one example of a class would be transcription factors. Right. So what are transcription factors and how do they differ from, say, like enzymes?
Right. So transcription factors are proteins that govern the activity of other genes. And some transcription factors, some of them may govern the activity of, 500 or 1,000 genes or more. So if you want to think in sort of hierarchical terms, use sort of any metaphor you want. These are sort of the generals and the other genes are sort of the soldiers, but a smaller number of genes, probably in the human, It might be on the order of 1,000 or so. So it still might be 5% of our genes encode transcription factors. But these transcription factors turn other genes on and off. Often they work in combinations. So some of the biological specificity and fine tuning comes from these transcription factors working in combinations. But if a transcription factor affects the activity of, say, 400 or 500 other genes. So let's say, for example, the body's building a muscle. There's a whole lot of stuff that you got to turn on to give that muscle its property of muscles, the fibers that are in the muscle, the way energy gets used by the muscle, the way the muscle recovers after exertion, the anatomy of the muscle. There's all sorts of stuff that has to go on. And so there's a muscle-specific genetic program, and there's a few transcription factors sort of in charge of that program. So those are really, hierarchically speaking, sort of, top of the hierarchy genes. And there are genes way, 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 we'll say, at the bottom of the hierarchy that essentially just carry out some job in the differentiated cell, the final muscle cell, some enzyme reaction or something like that cycles something in the muscle cell. And that doesn't influence other genes. It's carrying out an important job, but it's essentially at the terminal end of the circuit. It's the terminal end of the hierarchy. So however you like to think about it, I think hierarchies are probably a good way to think about it. You can imagine these transcription factors are so important because they do so many things. And then we realize they're so important. The easiest way to demonstrate that is to knock them out. So biologists have all sorts of tools for knocking genes out, whether that's, for example, in experimental animals like fruit flies or worms or mice. We can also do it in cells and culture and stuff. And we can show drastic, dramatic, sometimes catastrophic effects when these transcription factors are altered, because whole batteries of things don't happen when those transcription factors are altered.
Mm-hmm. So in startup speak, it would be like you've got some genes that are individual contributors, like they're doing day-to-day tasks. They know how to do that one thing, and that's kind of all they do wherever you find them. And then there's managers that are telling them where to go and when to do what they do. Right.
And some of these genes are kind of like executive vice presidents.
Yeah.
that actually control a lot of the managers. And there's a hierarchy to this logic. And when things get changed way up in the hierarchy, the biological impacts are dramatic.
Mm-hmm. I want to start talking a little bit about, keeping some of this stuff in mind, and we'll connect the dots for people. But I want to give people a sense for some of the basic principles by which animal bodies are put together. And, one of the things that you're going to teach us is, even when you talk about something like a fly compared to a human, remarkably, there are a lot of shared principles there. Yeah. But, let's think about flies and bugs and insects first, because when you look at them, their bodies are, there's literally segmented, right? Like you can think of a millipede. It's got a bunch of these almost like Lego blocks pieces put together. Where does this sort of segmentation and modularity that we see in something like an insect come from in terms of this genetic toolkit?
Well, it gets set up very, very early in the development of the insect embryo. I mean, the best studied embryo being the fruit fly embryo. But I think to start to paint a picture for listeners about this, I like to think, so a lot of embryos start out, they're spherical, they're close to spherical in shape. Sort of think about a globe. And I like to think about longitude and latitude and poles, right? So so think of something spherical. It's got a north and a south pole, right? It's got that equator, but along that there's all sorts of longitude, and of course there's all sorts of latitudes marked out north to south. As a body is built, essentially there has to be information as to what to build at all these positions. So at a certain degree, longitude and latitude, that might be where the eyes are going to go. At a certain degree, longitude and latitude, that may be where some appendages are going to go. And if you make this fully three-dimensional on the inside, that's where you're going to put, muscles and guts and all this other kind of stuff. So this three-dimensional spatial information is really important in development. And you also have the aspect of time. You're going to do some things before other things, right? It's almost like building a house. You realize you're going to lay the foundation before you, paint the drywall in the kids' room, right? Mm-hmm. Same thing in building an animal body. You're going to lay out some of the basic foundation, sort of this grid work, and then you're going to start filling in with specific body parts, specific organs in particular places. So segments are this very repeated pattern that lots of animals have. So insects belong to this group called arthropods. And if you think about, crabs and lobsters and other favorite things like that, or butterflies and millipedes, they have these segmented bodies. Well, we do too. The obvious part of our segmentation, if you look along our backbone, right? So we have cervical vertebrae, we have thoracic vertebrae, we have sacrolumbar vertebrae, et cetera. We are segmentally organized as well. So this, and so both animals, both insects and humans, have an organization of head to tail, right? We know the head goes in front. That's where the brains and the eyes go, right? And at the back end goes, where the waste comes out, right? So there's a lot of polarity. There's a lot of spatial information in a creature, and all that has to be set up. And so segmentation, this major feature that you can see so obviously in insects, but it's also there in us, that's going to get set up early because at different positions along that main segmented axis, that front to back axis of the animal, other things are going to form, ribs along your backbone, for example. All that information has to be set up. In an insect, the insect is laying out, there's three main parts of an insect. We all know it from either from biology class or from just looking under a magnifying glass. It's got a head, right? And that's got the mouth parts and the antennae and stuff like that on it. It's got a thorax. That's where the walking legs and the wings are, if it's a winged insect. And it's got an abdomen. And often that abdomen is segmented. It may or may not have anything sort of coming off of it. So in building an insect, you're sort of laying out head, thorax, abdomen, and then within that, a segmented pattern because the thorax has multiple segments, the abdomen has multiple segments, et cetera. So that sort of modularity, this is a really common feature in the animal kingdom, is that bodies are made of repeated bits. And then those repeated bits get specialized. So what gets built on the thorax of an insect, our wings go there, antennae go on the head. So there's a whole process for laying out the basic body plan and then for putting the various specific features in the right place on the developing body.
Mm-hmm. And I would imagine, when we think about the managers and the individual contributors in genetic terms, early on, right? There's got to be proteins in every cell from the beginning that do the most essential basic stuff. But then I imagine, right, there's some period of time as development is proceeding where you're eventually going to start specializing. You're going to make things like neurons and skin cells, and they're going to have special proteins that most of the other cells don't have. But it's going to take time to get those on. And in between, you've got some kind of cascading temporal logic by which these other transcription factor things are orchestrating those changes.
Right. So you've got transcription factors setting up these main body axes, the front to back and the top to bottom. So we know, for example, that, biologists will use the terms dorsal and ventral, but dorsal is our back, ventral is our stomach side, right? So different things happen on that part of our body from the other side of our body. Think about a deer, right? It's colored differently on its back than it is on its ventral surface, front to back. So these transcription factors are setting up these regions of the body, and certain transcription factors are responsible for what goes on in a particular region. Those managers are saying, okay, I'm part of the head program here, or I'm part of the thorax program here, or I'm part of the lumbar program here. And again, those things have to happen earlier and they have to go right, because when they go wrong early, you can imagine it's a cascade of disaster. Mm-hmm. So, essentially think about birth defects, the sort of birth defects we would probably never even see because things can be so catastrophic, it's inviable, right? Yeah. You don't build a viable animal when these things go wrong early.
Mm-hmm. So these, some of these key genes, these master regulators, that would mean that, if they mutate, it's catastrophic. Yeah. Actually, let me back up a second. So, we think about all of animal diversity, flies, worms, bugs, humans, birds, everything. They're all very different, but they've all got a head and tail. So there's that polarity there that you mentioned. They've all got some kind of body symmetry, left and right, top and bottom. All the stuff gets set up. Maybe, because I don't want to go too into the weeds and like the molecular details, but why don't I just ask you, like, let's take polarity. We all have a head and tail, all animals do. How does the embryo know which side the head is on?
Yeah. So it gets set up differently. It's sort of funny. Like there's not a universal rule for how it gets set up. In some animals, and say something like a fruit fly, there's information there in the egg. The egg's pretty big. And actually, so as the egg is developing in the mother, in the female, there's information being laid down. The way that cell is forming, there's information laying down that already essentially is specifying polarity in the cell. And the polarity in the cell becomes the polarity of the embryo. In other species, for example, where the sperm fertilizes the egg starts to set up that information. So the egg is kind of agnostic, and then where the point of sperm entry happens can set things in motion. So you need something to create an initial asymmetry.
Mm-hmm.
It may be built into the egg before it's fertilized. It may be once the egg is fertilized. But once you have that initial asymmetry and cells start dividing, that means you have different information across the egg. You have something that's localized. You have more of some stuff in one place than you have in another place. And that chemical difference can set in motion a whole cascade of things that can really then distinguish those different regions of the body from each other.
Mm-hmm.
I think hopefully that's a decent enough verbal description, again, without getting into molecular details. You need some kind of initial asymmetry that then cues a whole bunch of events that happen after that. Mm-hmm. And the molecules that are involved in these asymmetries, we've gotten our hands on all these things. They do spectacular things. We can manipulate them. So if we put those molecules in a different place, you can make some pretty freaky looking embryos. And that's a good way to test that you understand how things work. But that spatial information in the embryo is really what that means is there are chemicals in the form of proteins that are distributed asymmetrically early on. And those asymmetries are elaborated upon in building bodies.
So, maybe you could imagine a perfectly spherical egg, and it doesn't know which side is head, which side's tail. It's a perfect sphere, but the sperm enters at some point on that sphere. And maybe there's a chemical in the sperm, and it's highly concentrated where the sperm and the egg physically touch. And then there's a diffusion gradient that goes away from that point. And the head is like that point, and the tail is then the other point.
And it could also be something inside the egg that reacts to that and says, oh, okay, a change is now taking place at this point in the egg. That's going to be sort of my, North Star. That's going to orient the whole egg, and things are going to happen from there. So it could be, sort of a physical change that then triggers a chemical change, a cascade of chemical changes. So different animals use different mechanisms. It turns out there's kind of a variety of logic out there. Yeah, or a variety of mechanisms that's out there.
But I guess the principle here is some initial asymmetry is set up. That polarity then starts to get built in and elaborated on. And to put it in a very coarse way, you've got these cascades of different transcription factors and proteins that get turned on. And at different points along that embryo, they get turned on sort of in different combinations. And then ultimately, that's what's going to get you one segment being an arm versus a leg versus a whatever.
And people may start thinking, well, how do you make those distinctions? Well, these transcription factors also talk to each other, essentially. So some transcription factor, for example, may turn off other transcription factors. So you sort of get zones of exclusion. So you start setting up finer and finer. You start with very coarse. I guess the best way to describe it is coarse to fine delineation. So that you start with very coarse, okay, this is the front, this is the back, this is the top, this is the bottom. But that coarse sort of map becomes much more finer scaled as time unfolds. And it's all sorts of mechanisms, including sort of crosstalk between these transcription factors that are saying, hey, I'm active over here, stay out of my zone. And then that next thing is active in another zone. And it's talking, they can set up these, very restricted very restricted zones within the animal where different things are going to happen. Both along the main axis, including, into the body, because we also need to know I think there's a lot of songs about this from the 50s or whatever. You got to know, what goes on the outside and what goes on the inside, right? You want a gut running through the middle,
Mm-hmm.
not on the outside of the animal. You want, skin on the outside. You want to put a skeleton in the right place. So it's three-dimensional information.
Mm-hmm. And I would imagine, so like, as we start to think about like, okay, we get some sense for how the body is put together, and we've got, we can talk about transcription factors and the mechanisms that underlie that. Immediately, in as we're talking about this, you can start to intuit, I think, that okay, if I'm now going to start to think about evolution and think about how mutations tweak this developmental process to create a new type of animal, it becomes pretty clear and pretty intuitive, I think, that okay, if I mess with the sequence of one of these master regulators, that's probably going to be catastrophic. It's probably going to break everything because right from the beginning, things are going to go wrong.
Right.
And then that gets you thinking about constraints on in what type of genes and where on the gene can you start to get mutations that tweak what the animal looks like.
Right. So let's give some concrete examples about that sort of catastrophic argument. So, we know, either whether you want to think about humans or mice or fruit flies or whatever, we know what happens when these genes are rendered non-functional. And generally it's a very obvious and terminal effect.
Mm-hmm.
You could be missing whole parts of the body. You can be missing whole organ systems. You can be missing entire appendages, things like this. Okay. So that's what we mean by the scale of what can be disrupted when these things are missing. But when you think about, let's just think about, four-legged animals, and you think about the bodies that you see out there, and you think about, giraffes with their longer necks, or, something with maybe shorter limbs, or something with a longer body or a shorter body and things like this. You're like, they kind of all look like the same sort of animal's been bent through a funhouse mirror, right? Kind of elongating this or shortening that or whatever.
Mm-hmm.
So the kind of the basic outline is there, but what's changed is the proportions of things. And you have to do that in a way that's viable, right? These evolution is a matter of, you have to tinker while the engine's running, right? These things are not designed from the ground up like they build cars at Ford, right? You have to tinker while the engine's running. And what we appreciate now is that subtle changes in where these genes are turned on, how many cells they're turned on in, subtle changes in the subordinates that they regulate. These are the kind of changes that are happening in the course of evolution. Not in one step wholesale rearrangement. Those big changes generally aren't very viable, but subtle changes in size, subtle changes in the relative position of something. That's what you see happening. How do you do that? Well, you don't have to tinker with the transcription factor as a protein itself. You tinker with these subtle aspects of space and time. Well, I turn it on in a few more cells,
mm-hmm.
or I leave it on longer, or those cells divide longer. That's going to change the size and shape of something in a viable way. Mm-hmm. That's the sort of stuff that variation is made of. So what kinds of mutations have those properties? Those are generally mutations in the switches. Those are mutations in the non-coding parts of DNA that influence this choreography of how these genes work in space and time. And they're not catastrophic. And furthermore, these mutations, these switches, an individual gene, say one of these top regulator genes, it might itself have 10 or 20 switches associated with it. So, and a mutation in one switch has no effect on the other switches. Mm-hmm. So if you think about a gene, for example, that might be involved in building bone, it might have a switch that turns on very late, say, for example, in the elaboration of the fingers, okay? Well, tinker with that, and you might be changing the width and length of fingers, mm-hmm. okay? But you're not changing the backbone, you're not changing leg length or things like this, right? So it gives fine-tuning control over the proportions of body parts and the compositions of body parts. Yeah. So the evolutionary action in terms of the evolution of anatomy, the evolutionary action is in these switches that control this choreography. It's not actually in the proteins themselves. And that was we probably may need to get into that, maybe not, but that was sort of a breakthrough in thinking for evolutionary science, because until then, people were really thinking about how proteins change.
Yeah, and I like I like what the funhouse mirror analogy that you brought up. It reminds me, commonly in like a textbook, when you start talking about evolution and homology and how the basic structures are often shared across distantly related organisms, you might see the picture of the human arm and hand next to the whale's fin and the bat's wing.
Right.
And even as a even as a young student, you can appreciate, oh yeah, like it's the same basic set of pieces in the same geometric arrangement, but
Right.
this piece is longer in this creature and shorter in this creature. And it sounds like what you're saying is you can get there slowly, and little by little over time by just saying, okay, turn on this protein a little bit more or leave it on a little bit
Right.
earlier or whatever. And those are all coming from mutations in these non-coding switches.
Yes. Yes. Yeah. And whereas if you actually mess with the transcription factor, you might be missing the limb altogether.
Yeah, yeah. And that would also, I guess, kind of make sense of why so much of the genome isn't protein coding sequences. So you actually have a large palette to play with to do this fine tuning.
Right. And that's what's evolved. I think, let me just give you another example of sort of how to think about this. And of course, we're thinking about our bodies. We're most familiar with our bodies. How did we all change, et cetera? But the one way to drive home the role of these switches is think about a caterpillar and a butterfly. Okay. Same species, right? Now, we've all seen a beautiful monarch caterpillar. Pick your favorite butterfly, whatever. I'll just pick a monarch. Most people have seen them, right? That caterpillar is living a certain lifestyle as it crawls up that plant and it's feeding on that plant, and what it's eating, what it's doing, et cetera. Doesn't have wings yet, anything like that. And then it goes through this incredible transformations, metamorphosis into a butterfly, and migrates to Mexico, right? That's essentially two animals, built from the same genome. This caterpillar with an obvious segmented body, it even has little legs on the abdominal segments, on several of the abdominal segments, no wings. And then the beautiful butterfly with wings, that flies away. So same genome, same genes in that animal, but a certain program takes it so far to be a caterpillar, another program kicks in making the butterfly. That shows you the power of regulation. It's all the same DNA in that creature. And essentially, it's almost like you're getting two entirely different lifestyles out of one genome. And that's all about the choreography. So we don't have such, incredible transformations, but I think it's a very helpful way to think about how such different things can be built using the same set of genes. A caterpillar and a butterfly is probably the most dramatic thing I can think of, and probably the most visually appealing.
Mm-hmm. And it's easy to think about mutations in some of these master regulator genes that set up and orchestrate this whole developmental process. It's easy to imagine how mutating them can be catastrophic because everything downstream is affected. But are there ways in which they can actually lead to functional mutations that are dramatic? And here I'm thinking about, some of the stuff that you can tell us about in terms of the identity transformation of tissues, or how do we think about the fact that, okay, at some point, the centipede and the millipede actually got more segments. What's the genetic basis for actually changing the identity of something or creating more units?
Well, yeah, people are working on this. This is a pretty lively area of research. This is getting into some of those subtleties. But if you think, for example, about, you may not have appreciated just how different crustaceans are from each other, but if you like eating them, you can sort of appreciate, shrimp and lobsters and crabs are different, but they all have a really sort of some similarities in body design. But what makes a lot of them different is the number of different kinds of segments. What's devoted to how many segments carry legs, how many segments carry swimming appendages, et cetera. So clearly evolution is playing with the number and kinds of, for example, appendages in these creatures, right? The number of kinds and segments and what they bear. So somehow, while there's that commonality, a crab is a crab and it's not a lobster, right? So you got to go way back in time to when they had a common ancestor and realize they've gone their separate ways. So what we know is, as the embryo is being set up, there's a battery of about 9 genes, 8 or 9 genes that are specifying what's going to happen along that main body axis, the head, the thorax, the abdomen. And the territories they lay out can be subtly modified. So if you sort of think, I'm going to give, okay, here's another analogy. I hope everybody goes with it. I gave you the globe and longitude and latitude. Now it's Super Bowl Sunday. Think of a football field, all right? And you got the yard lines, the 100-yard lines, okay? Well, you can imagine that if, and just say, let's divide those up into the 10 yards divisions. That if you say, well, the 0 to 10 is different than 10 to 20, is different than 20 to 30, different than 30 to 40, et cetera. But now if, as you're setting up this field, there's an interaction that allows that 30 to 40 to spread to 42. Now 42 gets shortened to just 42 to 50. You've just changed the proportion between two segments, just a little tinker. Well, then what's going to happen in those segments is going to be different. And it's the territories that get set up that are occupied by these master regulators that are being tinkered with early in development. So by shifting the relative, I'll just call them territories of these genes, you can dramatically modify and sort of sculpt the animal to be different. Now, you don't get it overnight. Okay, we can do things in the laboratory and make really big changes, but that's kind of evolution can't deal with that. Like one of the most famous mutations in fruit flies is a mutation that transforms the antenna into legs. Okay, that's a nice little trick in the laboratory. You will not find flies in the wild with legs coming out of their head because they need those antennae to find their way in the world. Okay, but it shows that that's how we started to know that we were dealing with the stuff that governed what happened in any individual body part. But so when you just want to imagine how you get this sort of diversity, it's by tinkering with the layout of the body plans. Think of the body plan almost like a little bit of a blueprint of what's going to happen where you're tinkering with the layout of the body plans of these creatures. Mm-hmm. And if I gave you an example of things like arthropods, but maybe an obvious one in our group of animals, invertebrates, backboned animals. I mean, think about a snake. Mm-hmm. Some people don't like to think about snakes. I like to think about snakes. I look at snakes and I just see novelty all over the place. So they've lost their limbs. Snakes evolved from a limbed ancestor. So that tells you some big changes can happen, right? And we know it's happened many times in lizards independently, where legless lizards have evolved from limbed ancestors. All those are burrowing creatures. So a burrowing lifestyle is facilitated by losing these limbs. So you can obviously slip into nooks and crannies and things like that. So snakes have lost limbs relative to their ancestor. And we know, in fact, some of the very specific genetic changes that happen in the snake are in the switches that build limbs. So you didn't knock the genes out themselves. You changed the switches so that program for making the limbs did not happen. That's been clearly, shown experimentally. So that's a pretty big change when you think about limbed versus unlimbed. And, I certainly would have predicted it 25 years ago. And thankfully, the research has been done. I could say, yes, it's a change in the switches, shuts the limb program off, and that's how you get a limbless snake.
Yeah, and so, we've started to talk about mutations, and sometimes they can be catastrophic in the sense that the embryo is not viable. The final animal's never even built to even be tested out in the wild. Sometimes it might be built, but the animal just can't survive well. So if you get that,
it's ecologically not going to do well. Yeah.
Yeah. Like if you're the fly who's got the mutation that changes your antennae into legs, maybe you can run around for a little bit, but you're not going to be able to find food. And so you die. This now gets us to the concept of natural selection.
Right.
And I've got a bunch of questions here about sort of evolution per se, now that we've gotten a little bit of a background here in the developmental genetics. But let's just start out with the basics. What exactly is natural selection? And in your experience as an educator, what are some of the major misunderstandings that trip people up with natural selection?
I like to think of natural selection as really just being competition between forms. Okay? So that, and this is the way that the two great discoverers essentially of evolution, Darwin and Wallace, thought about it. They saw nature as a battlefield with competition going on out there, and those that would have a slight advantage for some reason, let's suppose they could run a little faster or had a few more offspring, they do better than other individuals. And all that means is that nature is the agent determining winners from losers. So let's just say winners from second place, third place, fourth place. It's not necessarily total losers, but it's a range. So it's a competitive process out there in terms of which forms do better than others, and do better means they might live longer and have more offspring, because it's essentially, if you don't have any young, it's a moot point because that's the end of the line. So that's natural selection is really about, the future generations descended from those individuals, doing well. So I think natural selection is best to think of as a competitive process. And what that means is it's favoring those things with a slight advantage, and it's disfavoring those things with a slight disadvantage. Well, however slight it might be. If it's a catastrophic disadvantage, it's no race at all.
And the currency for success here is simply reproductive output.
Yep.
Yeah, because you can live forever. You can be big and tough and strong and get away from predators. But if you don't leave the next generation behind you, then your lineage goes right away.
And those things that give you an advantage, they have to be heritable as well. So if all you've done is acquire wealth by building, say, real estate in New York, but your children are, never mind, I'm not going to go there. Okay. So anyway, so these are things that you acquire in life don't matter. It's what you're able to pass on through genes that matter. So that's natural selection. And I think the most common misconception is that in some way the organism is willing itself to that superior state, that advantageous state. So let me take like one of the simplest examples. One of my favorite examples is the story of the rock pocket mouse in the desert Southwest. So basically these mice generally occur in two coat colors, dark and light. And when you look at the distribution of those mice, when you go out and catch them in the field, most of the mice you find on dark rock. So this is in areas, for example, where there's been a lot of volcanic activity. There's dark lava rock. You find dark mice on the dark rock, and you find light mice on the like sandy rock or sandy desert floor. And that pattern of distribution is actually due to the activity of predators. The mice have no idea what color they are. Okay, but visual predators, owls, snakes, etc. When you have a color mismatch, those mice stick out and they're picked off. So essentially, it's the action of natural selection by the predators that is shaping the distribution of these creatures. And you think, okay, being dark on dark rock gives you an advantage. In fact, immeasurably much large advantage, selective advantage. Being light on light background gives you a strong advantage. But the mice have no influence over the mutational process that makes them light or dark. That is entirely a random throw of the genetic dice. This is the hardest thing for people to get about natural selection, is the creature has no agency in what's going to happen. Mutations happen at random. They affect body characteristics at random, and whether or not that's advantageous or disadvantageous depends upon the conditions in which the animal finds itself. So if you're a light-colored mouse and you happen to pick up a mutation that makes you dark, that's advantageous if you happen to stroll over onto some dark rock. It is disadvantageous if you stay on the light-colored sandy soil. And you can think vice versa. It's the conditions that you're living under. And if you think, oh, well, up on that dark rock, there's that lava rock as it breaks down, it's a nice substrate for plants to grow. It's richer, and there'll be more food up there in the rock, and I want to get up on that food. I need a dark mutation. The mouse has no agency in becoming darker. Whether it's dark is entirely dependent upon things that happen in DNA that are not under its influence. So this sense of agency that somehow creatures can, will or drive themselves to adapt, it's probably understandable why our brains might think that way, because we'd like to modify our behavior to do that, but that's just not the way nature works. The genetic lottery is random, and whether or not something's advantageous or disadvantageous depends upon the conditions that creatures find themselves.
And so, yeah, the conditions, the natural world that the animal's embedded in is what's doing the selecting. That's where the agency is, so to speak. And what it's selecting for are forms that are able to survive long enough to reproduce.
Right.
So the light mouse on the dark Ravalok is going to get picked off before it has a chance, probably, to reproduce.
Right.
But then the other thing that comes into play here that's interesting is the sort of reproductive act itself. So the interaction of, say, the males and females of a given species. And here's where you start to talk about sexual selection. So let's just start with a simple definition. What is sexual selection, and how is it similar to and different from natural selection?
So sexual selection, again, another Darwin insight, is that there's all sorts of characteristics that seem to affect reproductive success. And they could be, for example, things used most obviously would be in things involved in courtship display. So the idea would be that, we can think of all sorts of species where males are in competition with other males. They're trying to get the attention of females. And so whether it's, a dance, or whether it's the size of their horns, or whether it's a chemical musk they put on themselves or whatever, there's all sorts of competition out there for a mate. Very, very often, male competition for a female, and the female's making the decision as to what's going to happen. Pretty common in the animal kingdom. And those traits under sexual selection, boy, they can evolve pretty rapidly, and they can get very, very, as Darwin would call them, sort of exaggerated, right? I mean, the peacock's tail. I mean, holy smoke, right? I mean, what an extravagance, right? Some of these horns, right, on, or antlers on animals. I mean, ridiculous size. It takes a lot of energy just to build those things and to hold the neck up, et cetera. But that's because the mating game is where a lot of the action's at. So as you're sort of dialing in here, we talk about natural selection. Sexual selection is essentially a form of natural selection, but it helps us sort of focus on the mating game, on the reproductive contest. And, when you think about the structure of all sorts of things, where a male might have, 15 or 20 females in a harem and competing with other males, that's a big competition as to whether or not that male's genes are spread, make it into the next generation. And this competition that's been going on probably for as long as the animal kingdom's been in existence.
When I think about the pocket mouse example with the light and the dark rock environments, that one makes sort of complete sense. Okay, the dark mouse is going to blend into the dark background. The light mouse is going to blend into the light background. This enables them to evade predators long enough to be successful in the rest of their life and find a mate. When I think about sexual selection, sometimes I also get a sort of complete feeling to my sense of understanding as to why these ornate structures evolve, but sometimes I don't. So let's look at an example of each. And this is, by the way, this is a genuine question I have. I don't actually know if this is in the literature. Anyone's got a good explanation for this one. If I look at something like a white-tailed deer, and I think about the males competing with their antlers, if I just go, okay, I accept that the female wants the one that's going to win the fight, it makes sense for why they're going to have bones that grow out of their head, and the one with the bigger, better antlers is going to win the fight. That one's intuitive. Even though it's hard, it's heavy to carry around, it's serving a clear purpose, and that's a literal battle. When I think about something like a peacock's tail, it's from the outside looking in, I see a hindrance to survival. The animal's going to be slower. It's going to be harder to fly. My question is really, why do we know anything about where that female preference actually comes from? What are the females selecting for that's going to help their offspring in a case like that?
Well, I don't really know that if we have really, watertight evidence. I think the thinking is that females are generally dialing in on these characteristics as some sign of, for example, male health. So it may be simpler when we think about, say, for example, certain birds that might have head feathers. And some of these colors in birds the birds can see in the ultraviolet. They're actually colors that we may not be able to perceive, but things like zebra finches and stuff like this, it's been well studied that it will reflect to some degree, male health,
Mm-hmm.
the overall condition of the male. And that sort of reflects, now again, this will come in for the rationale, that sort of reflects that it's been able to access food. It's doesn't have any obvious body defects, things like this. So in some ways, it's, that showiness is a way of saying, I'm so well fed, I'm so well traveled, essentially, that I built this magnificent tail, and oh, don't you want a piece of my genetic material?
Mm-hmm.
That's sort of the rationale. There's trade-offs, though. What you're talking about is exactly, it's invested energy in making this tail. It's in the case of the animals locking horns, there's not only just the energy of building the horns, but of course there's tremendous energy involved in combat and things like this. And of course, as you know, a lot of males in a lot of species die younger,
Mm-hmm.
right? I mean, male lions, same thing, right? Then they get kicked out of the pride, they go off and die by themselves, etc. So you can have a lot of fun with this. Sort of looking at the males is essentially, just sperm on 2 or 4 legs. And, as long as the job gets done, it doesn't really happen. It doesn't matter what really happens to the male. So, the male's job is, or, sort of think about it, the male's job is to procreate. And these are essentially these flashy, sort of almost luxury items that's evolved to sort of be, symbols of success, symbols of health, surrogates. Because you can't really, the female can't know that male's got great sperm.
It's potentially like a genuine display of what you might otherwise fake. So if you've got the biological resources to make the peacock's tail, it says that, well, you've obviously been around long enough to be able to allocate and acquire resources enough that you've got a bunch left over to make these cool feathers.
Yeah. And there's a trade-off. I mean, so often in biology, there, we can see there's a trade-off. You've invested resources there. It may make you slower. It may make it easier for some predator to find you and track you down, et cetera. But in this trade-off, the mating game, the imperatives of the mating game are outweighing the drawback of, being grabbed by a leopard or whatever it might be.
Mm-hmm. I also want to ask you about, when we talk about evolution, we were just talking about how mutations are random. The animals aren't choosing or willing their adaptations into existence. It really is a genetic lottery. You can't control which base pairs are going to mutate generation to generation. And yet, we've talked about some important constraints here, like the catastrophe that comes from mutating certain genes in certain places means that variation sort of never even starts the race. It never even sort of enters the picture. Other mutations you can play around with more. So there's constraints on where mutations can accumulate in the genome. And there's also the phenomenon, say, of convergent evolution, where, obviously evolution is somehow coming to the same kind of adaptation over and over and over again in a really repeatable way.
Yeah.
And so my question is, with this element of random mutation, is there a predictability to the evolutionary process, and how predictable is it?
Well, that's a superb question. I think we're definitely getting down in the kind of some of these sort of beautiful black boxes that only recently I think we can sort of shed light on. So we can't predict where a new mutation is going to happen in an individual. These are distributed throughout the genome. It's just a probability process. But given a sizable population, knowing that individuals are born with new mutations, we can estimate that, for example, give me 50,000 newly born, mice from sandy parents, I might see a black offspring in there, right? Because I know how many genes can give me a black fur coat. I know how many mutations might be able to give me that. So I can kind of come up with the math of this. This might be a 1 in 50,000 event. This might be a 1 in 10,000 event, et cetera. So we can at least predict sort of a probability that something might happen. But give me the conditions. So, for example, give me hundreds of square miles of desert where over the last 1,000 or 2,000 years, there's been all sorts of eruptions and lava flows. let me go in and look at the mouse populations on those black rocks. What I'll find is over in one part, I'll find certain mutation that makes those mice black. And I'll go to another population and I might find a different mutation that made those mice black. So they've arrived at the same solution, sort of the imperative of their conditions, right? The conditions have said dark mice are going to do better on dark rocks. They may or may not have gotten there through the same mutation. Kind of depends on the nature of the trait that you're talking about, of how easy or how likely is it for the same mutations to happen.
But there's probably a lot of traits for which there are many mutational paths that can get you to the same outcome.
Yeah, I think path is a great way to use that. You almost sort of think like, actually, this goes almost probably a century back in evolutionary thinking, that if you think about, a mountain range and you're going to, climb that mountain, how many paths are there up that mountain? How many different trails could you possibly take? If you're trying to get to the same place, which is sort of peak fitness, dark mice on dark rock, and there's more than one way up, nature will find a way to take more than one way up. Now, if one way up is many, many steps, and the steps in between are no advantage, it won't take those long ways. It won't take those long and complex routes. It still needs a route that gets you there. But it's beautifully satisfying. Both cases where we see the same mutation happen independently, many times over, where we see maybe different mutations in the same gene happen independently, but also sometimes we see essentially species execute the same advantage or achieve the same advantage in almost entirely different ways. I see lots of these examples in snakes, where actually just think about animals, for example, that snakes have to get their toxins into your bloodstream. Okay, well, there's all sorts of groups of snakes across the world that have evolved over time, and it turns out that they're often messing with your blood coagulation. There's more than one way to mess with blood coagulation, but sometimes snakes have independently come up upon essentially the same solution. There's ways to make your blood pressure go down. There's independent ways to come up with that, and there's sometimes independent inventions that use the same way. And then think about blood-feeding animals. Think about ticks and vampire bats and things like this. They want to keep your blood flowing when they're sucking, right? Or leeches, right? So totally different groups of animals feeding on, say, for example, a mammal, and they've got to put enzymes in there that prevent your blood from coagulating so that they can keep the blood meal going. So when we look at strategies like that, ecological strategies, I'm a snake, I want to get a toxin through your bloodstream, or I'm a vampire bat, I want to feed on your blood. These are lovely topics, I'm sure that your audience is enjoying. But rest assured that when we start to understand what's been tinkered with in evolution, we can often see similar, if not identical, solutions being come up with by different animals. And that tells us how strong the conditions of selection are, that things will arrive at similar or identical solutions given similar conditions of selection.
I see. One of the more interesting concepts that I was introduced to in some of your writing was, classically, before the dawn of molecular biology, if you wanted to understand evolution and natural history, you had to look at physical macroscopic things, like go dig bones out of the dirt and go, oh, wow, there used to be these things called dinosaurs and we can see their bones. Looking at things at that scale. But after the molecular revolution, you could look at genomes. You could look at the actual molecules, the base pairs of DNA and stuff inside of organisms, and you could go fossil hunting in the genome, so to speak. And one of the coolest concepts that I read in one of your books was the idea of fossil genes, that you can actually see the evidence of evolution by looking at the genome of an organism and saying, like, okay, this animal once had this, that, or the other protein. Right. How do fossil genes work, and what are maybe one or two examples of this type of thing?
Yeah, this is. I'm glad you appreciate it. I always thought it was so cool when we started to have this power, and to look into genomes and then realize there was a history in there. And I'll take my favorite example is this. So every animal with a backbone on the planet has red blood cells that use to carry oxygen, except a little group of fish in the Southern Ocean off the shelf of Antarctica called ice fish that are transparent and have no red blood cells. Okay. So it's probably no more than like a dozen or so species. All right. So of the millions and millions of species of vertebrates that have ever lived on this planet in the last 400 plus million years, they're the only ones we know of that have gotten rid of red blood cells. And we know we got rid of them because we have all sorts of red-blooded relatives we know of these ice fish. But when we look in the ice fish DNA, we can see the genes for the protein that carries oxygen, the genes for hemoglobin, are fossils. The code is there, pieces of the code are there, but it's broken. It's screwed up. And so what these fossil genes are is they're like genetic text where we can recognize, hey, it's like you've got that stretch of DNA, if you think about text, it's like you've got the page from the book there, but there's typos and deletions and stuff like that. And it says at one point, it had an ancestor that had intact text, and we can infer that from other species, but it's decayed now. Its mutation has started to erase that gene in these animals. We've got hundreds. We got some significant number of fossil genes in us. A lot of species are carrying this around because they've shifted lifestyles. A fish that used to live an ancestor who lived in temperate waters, relied on hemoglobin to carry oxygen. It turns out that there's probably a disadvantage to having red blood cells when you're living at almost -2 degrees Celsius in those ocean waters. And in fact, also there may be iron is limiting, and you need iron for hemoglobin to work. And so you don't have a lot of iron in the diet. It's a liability. Selection has favored the animals that have lost the red blood cells. And when you look in their DNA, they can no longer make this hemoglobin, which is just, it's a fossilized gene in there. It's almost intact, but it's got enough breaks to it that it tells us no longer operational. But essentially, it's like a vestige of ancestral life. Mm-hmm. Still tells you a lot about the ancestor. And we've got these in our genome, and all sorts of species have them all over their genomes. That it's another way to look back in time and say, well, that was a piece of code that worked in some ancestors and still may be working today in some close relatives. But in this lineage, in this group of creatures, no longer working because something's changed about its conditions of life. And now this thing is either not needed or it's a liability.
Mm-hmm. So you mentioned that we have a lot of, I imagine every species is going to have some fossil genes because they've all got some history. Humans have fossil genes. We have a number of them. Are there any patterns to the fossil genes that we have? Are there any themes, like certain classes of proteins that we've lost?
We've got a big number of fossilized olfactory receptors. Okay, so here's the thinking about that. So one of the biggest gene families we know about in mammals encode proteins that help us detect odors. And it's a very rich family. And if you look at things like mice or dogs, I think it may be close to 1,000 members in the family. But you look at humans, hundreds of them are fossilized. What's going on? Well, the best way we can rationalize this is, we're great apes. Our closest relatives are the old world great apes. That's the only group of mammals on this planet that has full color vision. So our ancestors evolved full color vision. Full color vision, we actually, and you know this because you know neurobiology, that we've devoted a fair amount of our brain to our visual system. Mm-hmm. And we navigate the world in color. And we make maps of the world in color. And we identify foods to eat or foods not to eat, using color that other mammals can't. So the thinking is that we've shifted to a more visual. We're relying more on our visual senses to make our way in the world than, say, imagine a mouse scurrying around the ground that can't see very far ahead, and is finding its way in the world using its sense of smell. So that's a case where a lifestyle change, becoming more visual, meant we've left behind a fair amount of the olfactory system, and we also can see genes that are involved in building some of the anatomy of our olfactory system are fossilized genes. So we've discarded a bit of the olfactory capabilities of our ancestors, living a more visual lifestyle. But when we look at other mammals living a very olfactory lifestyle, don't have color vision, those genes are intact. Kind of a beautiful story, isn't it?
Yeah, I mean, it all fits together very nicely, and it really does make a lot of sense. Obviously, we still use the sense of smell,
Yeah.
but I mean, you can just intuit,
Yeah.
in a common sense terms. We don't use our smell like a dog or a bear does, and it's very, very easy to imagine, based on what you just said, that although we can smell a lot of smells, a lot of odors, the repertoire of potential smells is probably more limited for us than it is to them, analogous to the way that their color spectrum is more limited in terms of what
That's
they can
right.
see.
So we can detect all sorts of hues of color that they can't, and they can detect all sorts of ranges of smells that we can't, which is why you got all those dogs in the airport.
I want to ask you about the speed of evolution. Normally when you talk about evolution, you're talking about very, very large stretches of time, millions of years to get new species and things like this. Thinking about the dinosaurs and very, very, very long stretches of time. The question I essentially want to ask you is, and I think I would like to use humans here as an example, but there's probably lots of other examples we can use. How fast can evolution work? What is the speed of evolution or the speed limit of evolution? And what are maybe some examples of the more recently evolved things that have sprung up in human beings that help us get that speed limit?
That's a great question. I still think we're all wrestling with this. And we need examples, to learn from. But I'll start with human, and then I may just kind of then bounce around a little bit to sort of just spur some thinking, maybe some visual examples that might occur in listeners' heads. Well, if you look at a human, the most impressive thing of a modern human relative to some of our bygone ancestors is brain size. Mm-hmm. It's not overrated. It's maybe underused, but it's not overrated. So from the period of about 2.5 million years ago to, let's say, maybe 1 million to 800,000 years ago, brain volume in humans tripled in size. That is a spectacular change. When you take into account what that means in terms of capability, all that neural capacity, when you take into account how much energy it takes to run the brain, that's a pretty big commitment to that. Now, you might say threefold increase in size in 1.5 million years. Maybe that's, for a brain that may not be that impressive. I can't tell you if I was trying to say, how much has like horse body size changed per 1 million years, or do we have a good sense of how much whales changed in a few million? Gigantism in whales, I think, is a relatively recent phenomenon. I think some people maybe have some kind of peg on the time there. So I don't want people to go away from this and go, oh, Sean thinks threefold is spectacular and it's the top of it. But it's impressive because of the body structure we're dealing with, mm-hmm. and obviously all the impact it has on lifestyle. And not only is the amount of, you want to know about the speed limit of evolution, and I can't tell you whether the brain could have evolved any faster than that. But the why the brain probably evolved in that period is really interesting, which is we're in an ice age. Mm. I understand it, the first ice age in 300 million years on the planet. Ice ages are a really weird time. Ever since the asteroid hit the Earth 66 billion years ago, the Earth has been pretty warm. And in fact, for millions of years after that asteroid impact, the Earth was green from pole to pole. You did not have any ice at the poles. You only started getting ice, for example, at the South Pole probably 30 some odd million years ago. And then the freezing over of the northern hemisphere, and then the advance of the ice sheets, and then the retreat of the ice sheets, and all that kind of stuff, that kicked in 2.5 million years ago. In Africa, it's not so much a story of cold and warm, it's a story of wet and dry. And our ancestors, by 2.5 million years ago, had invented tool use. So stone tools, primitive tools. But if you look at the next 1.5 million years, that stone toolkit got much more sophisticated. Somewhere in there, maybe on the more recent side of it, came the use of fire. Fire obviously allowed us to control some of our habitat, allowed us to cook our food. You get more calories out of it. We became a technological ape, essentially, in that 1.5 million years or so. And, quite obviously, we're still technological apes. So that brain size increase, it was profound for our lineage. Obviously, it's been profound for the planet. And the closest causal link we could say is it's the ice age, and that you're living under these regimes of shifting climate, wet and dry. And the ape that can control its habitat, the ape that has a little more cognitive ability in terms of finding food or whatever it might be, doing a little bit better.
Mm-hmm.
And
so
the big-brained apes did a lot better.
Yeah. So basically, you can go in terms of sort of a gross anatomical change, and a very, very important one that basically defines our species. You can go from something about the size or not much bigger than a chimpanzee brain to a modern day human brain in about 1 million years.
Yeah.
Maybe you could do it faster, but that's how long it actually took in our lineage. What about what's like an example of like something that's happened so recently in evolutionary terms that it hasn't actually spread to everyone yet? An example that I can think of here would be like the lactase persistence story.
Yeah. So I think, to my knowledge, the ability to utilize the milk sugar lactose evolved twice in humanity. I'm not totally up to date, Nick, on the literature. I'm thinking this is in the last 6,000 to 10,000 years. So it's going to correlate with the use of livestock and obviously harvesting, milk from livestock. Mm-hmm. Hasn't spread to everyone. That's why some of us are lactose intolerant. But that's a great example of a trait that clearly came along, I think, with civilization and domestication of livestock. Hasn't spread throughout the population all the way.
But we're talking like thousands of years. In a matter of a few thousand years.
There's a few thousand years.
You can see a change like that spread to a lot of the population.
Yeah. I also think somebody told me, we don't think there are many. Well, hey, just look at skin color. Okay. Mm-hmm. So probably the big migration out of Africa that's peopled the world probably started 60,000 years ago. Mm-hmm. All of us who are fair-skinned, who are light-skinned, that's a big change from our African ancestors. It's thought that has to do with being in northern climates, that you have to have less melanin in order for vitamin D to enter the skin in the way we need it. We rely on it for a lot of body functions. So that change in pigmentation is going to be within the last X thousand years. And you can also see that different grades of pigmentation exist in humans, across the world relative to latitude and altitude. So you can, those are some examples of traits that have definitely changed in geologically recent humanity. Yeah.
Mm-hmm. I mean, I haven't followed this area of science, as closely as I used to. What are one or two things going on in evolutionary biology right now where people are making progress and we're probably going to get some answers to some interesting questions we didn't know about before?
I think probably the things that people hear about and that are interesting are essentially paleogenetics. So, this is where archaeology now has met genetics, and our ability, which, didn't start till, late 80s, early 90s, that we started getting DNA out of museum specimens and starting to analyze it. But this has ratcheted up to, phenomenal high resolution, high speed work. And the ability to, look at specimens, thousands, many, many thousands of years old, both human pathogens that are associated with humans, livestock, et cetera. This, I think, is, archaeology and anthropology are in both in a renaissance and perhaps, sometimes an uncomfortable revolution, because ideas that have been around for a while are being tested with new methodology. But whether you're interested in, plagues that swept humanity and may have, changed the course of history in Europe, or changed, in fact, since the ancestry of humans, because you had some plagues that are pretty large scale and had a pretty big impact on who the survivors were, understanding the origin of pathogens. I think that's an area in sort of a human evolutionary studies that is on fire in terms of the amount of work going on and the potential there for, I think, just that findings that help us stitch together our own history. I'd say particularly from the last 10,000 years. I think everybody's familiar. This has been going for a while. The genealogy studies have been there. Where did you come from? We're a country, obviously, of immigrants, and everyone's interested in where their ancestry goes. But we're talking about a little deeper time, a little more complex changes, adoptions of various technology, who introduced which animals where, which crops where, what's the story of humanity of the last 10,000 years? And I think that, and people are, going through graveyards and bogs and everywhere to try to get specimens to tell that story.
Interesting. Yeah. So just piecing together a story of, there's a story of deep human history. So like the Svante Pabos of the world looking at, Neanderthals and ancient extinct lineages of humans. Right. But there's also like the more modern historical record of how did people move around in Europe and bring sheep to this place and that place and all that stuff.
Yeah. And, who domesticated horses and all that sort of stuff. So I think the last 10,000 years where we know this is where crops were domesticated, livestock were domesticated. We imagine that the people that were doing that had interesting advantages in terms of, food resources. And then, of course, you had just waves of people moving around the globe and not with, great abandon. It did depend on modes of transportation at some point. But, and of course, these events where, human population was much smaller. You can imagine that some of these infectious diseases that we may have encountered as we entered new areas may have, devastated human populations. So I think the story of the last 10,000 years is going to be, the synthesis of paleogenetics and archaeology and anthropology is underway. And I don't know if the dust will ever settle. But for a while here, I just think it's a really fertile period.
Is another interesting question I have for you is, what is the relevance of the study of evolution beyond just the academic interest we have in knowing the story of ourselves? In particular, is there any advantage that would be had by, say, a medical student or a physician in understanding something about human evolution and evolutionary biology that would actually enable them to be a better healthcare provider?
Yeah, I think so. In fact, there's this whole realm called evolutionary medicine, and there's groups that get together across the nation who get together on, for essentially Zoom seminars to try to sort of use evolutionary knowledge from a medical perspective. I'll take the low-hanging fruit here. Okay. We had this pandemic. Did you hear about this? Last couple of years ago, et cetera. Everything about how that virus was evolving, and it's evolving under the intense selective pressure of our immune systems, both our immune systems as it responds to infection and our immune systems as it's responding to vaccination. And it was really important to try to figure out just, how far and fast could COVID evolve and in what directions and still be COVID, right? And, still be essentially, dangerous. And so, scientists, for example, were studying the spike protein that's on the surface of the virus. That's what's in the vaccine. So to understand the arms race, for evolutionary biologists, I think some of the most interesting situations are what we would describe as these arms races. And that arms race is describing, it's predator prey, it's host pathogen, these examples. It's where evolution is to use another metaphor, is on steroids. It's essentially, it's in very fast motion.
Yeah, because there's such strong selection.
There's a counterstrike. And in the case of a virus, it's got a very rapid replication cycle. So it's spitting out variants, in every individual.
Yep.
Multiply that by the tens of millions of people it's infecting, and you're getting a huge experiment going on with this virus, right? So arms races are just really interesting. That's a different way to put the COVID story. But what we're trying to learn, you remember you were talking about which mutations were viable, right? What's the mutational path? So the virus, for example, it's mutating. As it mutates, it's spitting out all these variants. Remember, at random. It's just essentially, you just have all these viral variants. It's spitting them out. Our immune system is clearing out the ones it can recognize. So, of course, the ones that it can't recognize, because it hasn't seen it before, those are the escapers that are then, seeding the new variant that we're all, worried about. But this process, the virus still has to be able to infect cells. It still has to be able to recognize the protein on human cells that it uses to get into our cells. So it's changing the spike protein under the pressure of our immune system, but it can only change it in certain ways and still get inside our cells. Right? So this is the things that we've been talking about in this conversation. What mutations are advantageous? What mutational pathways are permissible to the virus? What things won't work because it will cripple its ability to infect cells, game over. And when this is run for years against the immunity of the globe, how many more tricks does COVID have, right? So we hope it's kind of exhausted itself in terms of what it can come up with that our immune system hasn't seen yet. You were just asking, why would a knowledge of evolution be helpful? HIV was a totally different story. Some of, people may be too young to kind of remember all that, but HIV has an incredible mutation rate. Perhaps 10,000 times the mutation rate of, something like our own cells. So that means in an individual infected with HIV, that individual has a whole spectrum of HIV variants. That's why and then the insidious thing about the HIV virus is it attacks the immune system. It attacks the very system that we use to counteract the virus. So that arms race, and that's why it's such a deadly virus, is that arms race is as that virus essentially escapes the immune system, it's destroying it. And so the capability, obviously, of the infected person to respond to subsequent waves of variants is thwarted. So, our battle with infectious agents, and take malaria in Africa or whatever, these are large-scale public health battles. And I think to understand mutation, variation, the pathways of mutation, our adaptive immune system, people may not appreciate that, but the way our immune system reacts to these things, we actually rely on a mutation and selection mechanism in our immune system. This could be another 15 minutes of this podcast that people don't want to hear, but our immune system is amazing because, in fact, our countermeasure is to generate all sorts of immune molecules using mutation, somatic mutation. Now, this mutation only takes place in our immune cells to generate all sorts of variants to try to counterpunch the variation we're experiencing from a pathogen.
You're talking about the way our bodies produce antibodies.
Yeah, the way we produce antibodies. And people may not appreciate that what we do in making antibodies, our body can make billions of different antibodies using only a few hundred genes. So I repeat that again, because everything we've been talking about is like a gene encodes a protein, right? But the way antibodies are stitched together, and because of certain mechanisms that are particular to antibodies, is that using just a few hundred pieces of antibody genes that can be assembled in all sorts of different ways, the body can make billions of different kinds of antibodies. And for that, we should be thankful, which is why essentially we will mount an immune response against virtually anything we're exposed to, right? And that's the story of our group of vertebrates, is that 400 million years of vertebrate evolution has given us a pretty impressive immune system, which is a a very alert, very responsive system for counterattacking foreign agents that we've never seen before in the body. It's a really dramatic. I mean, you talk about it, what should a physician understand about evolution? Well, when you understand that our own adaptive immune system runs on the principles of evolution in a fight against all the infectious agents that could ever exist in the world, including new ones that are evolving as we speak, I think a knowledge of evolution seems really sensible to have if you're going to go into the field of medicine.
I do want to ask you a little bit about the snake venom stuff, just because it's so interesting. I don't think you quite said this, but I was detecting that maybe snake venom has evolved independently in different lineages multiple times. Can you maybe give a survey of the different types of venom and what we know about what the venoms actually are?
Certain toxins. So different groups of snakes have come up with some different toxins. But so snake venom is old. It may be, it's very likely that essentially lizards came up with venom, meaning something in a secretion that was delivered specifically into prey and to help immobilize the prey. But different groups of snakes have kind of gone wild with this idea. So if we work on rattlesnakes, they're easily accessible to us, maybe hopefully, familiar to listeners, hopefully not too familiar to listeners. Their close relatives in this country are copperheads and water moccasins. They belong to the same large family. Those snakes are largely, very often hemorrhagic in their nature. So they disrupt hemostasis. So they cause hemorrhaging, cause a drop in blood pressure, a lot of tissue damage. When they bite humans, they're very effective at subduing prey. And this is, obviously venom is first about subduing prey. When we get envenomated by a snake, it has no intention of eating us. That's a defensive bite. But there are rattlesnakes that are very neurotoxic and that will cause respiratory arrest very quickly. So particularly in the desert Southwest, things like the Mojave rattlesnake are something to be alert to. There's also some populations of snakes, even here in the East, that also have neurotoxic capability. Looking around the world, probably the Australian snakes are most famous for this. Maybe that's partly biological reality, partly Steve Irwin. So the snakes we have here that I just described are called pit vipers. There's snakes called elapids in Australia. The most toxic venom we know of in a snake is in an Australian snake called a taipan. Interestingly, depending upon its prey, a taipan can kill in different ways, but one way it kills is essentially exhausting your coagulation system very quickly. And it's come up with toxins that are in fact components of the blood coagulation system that it has co-opted into the venom gland. So it injects those into its prey and wrecks its clotting system.
I see. So it's almost like taking its own blood clotting proteins, scrambling them, and then injecting that into another creature.
Yeah, not even that scrambled. Yeah. It's essentially saying that there are certain physiological proteins, things that control blood pressure, things that, for example, control clotting.
Yeah.
That when injected into prey in some quantity.
Yeah. If it's concentrated, it's just at a dose that's super physiological.
It's toxic. Yeah. It's toxic. So something that has a normal physiological function, working in harmony with everything else in the snake's body, put in a venom gland, deliver it, and it's a payload into prey. Wow. And that's essentially giving away, for example, probably the main concept here is that when I said, well, where do these toxins come from? They're coming from normal physiological proteins that it just turns out some subset of them, when expressed in the venom gland and delivered through that hypodermic needle that is a fang, subdue prey. And it's an arms race with prey, because prey themselves have certain defenses. You could select for prey that have certain countermeasures. Some of those countermeasures might be things in their bloodstream that will soak up toxins. It might be variation in some of the things that the toxin hits, say, for example, parts of the nervous system. So we know that this predator-prey relationship between snake and prey is one of these evolution arms races that's going on. So that's part of the reason why we wanted to study it. Evolutionary biologists like to study things that have changed. So snakes have come up with new weapons, and prey have come up with countermeasures. And that's a rich area for understanding, this dimension of evolution, the origin of novelty and coevolution of venom and anti-venom measures.
Mm-hmm. How long ago? A lot of what we were talking about in the beginning, that was me kind of pulling up our past, because I read your book, Endless Forms Most Beautiful, when I was in high school. That came out in what, 2005?
Yeah. I was a much younger man, Nick. Yeah.
Well, so was I. Sean is one of the few living people on planet Earth who knows what I look like without facial hair. Fun fact.
Yeah. There's very few people on planet Earth who know what I look like without facial hair, because I've had it since I'm 18. And I'm a lot older than you now, buddy. Yeah.
And where are you at now? University of
Maryland.
Maryland. Okay. Yeah. And so are you focused mostly on the snake venom stuff now? Are you still doing fly work?
No, just in the lab moved here from the University of Wisconsin about 6 years ago. And I thought I should take on new things. I've done that in the research lab over the course of, oh goodness, now 36 or 37 years and leading a research team. I felt satisfied with what we had done in developmental and understanding body part development. I felt satisfied with what we had contributed to understanding the evolution of form. And I thought, let's get to biochemical novelty. Let's get what's going on. And the idea is not so much to solve the snake mysteries unto themselves and say, here's what snakes did. It's the general question of how does novelty arise? Mm-hmm. How does a snake come up with, put a clotting protein into its venom? What are those genetic steps? What's permissible? Why those proteins and not others? So it's understanding more of the general logic and rules surrounding the evolution of novelty, using snakes because it's just a rich set of examples that we can get at, right? And we understand the ecological significance of what's been invented.
All right, Sean, I don't want to take too much more of your time. We've already talked about a lot of interesting areas. Is there anything that you want to reiterate or go back over from what we said, or just sort of hammer home for people in terms of, how they should think about evolution, or maybe where they can go to learn more about some of the stuff we were describing?
Well, if they're interested in a little more of how this stuff works, you've mentioned Endless Forms Most Beautiful. There's also a book I wrote called The Making of the Fittest. But I'm gonna take that door you just opened, and I'll just say what's on my mind when I talk to people about evolution these days. And I gave you a little piece of it, and I'm going to throw this out there, and I'll just see if any of it sticks, and maybe a listener can offer a comment. I think we kind of need a new origin story. And if you take an evolutionary biology class, or gosh knows what you might be exposed to, you're going to start 4.5 billion years ago, et cetera, et cetera, et cetera. I really think when you look around the planet, I think the most significant event I can think about is that asteroid that hit 66 million years ago. That was a reset button, and it so changed the world, changed it from the age of reptiles, opened the door to the age of mammals. Our ancestors, mammals, took off. They got bigger than they'd ever been very quickly. They'd been around 100 million years alongside the dinosaurs. They got bigger than they'd ever been. All these groups we know and love of the different kinds of mammals evolved in probably a pretty short period of time, maybe 10 million years after the asteroid. But 3/4 of the species of the planet were wiped out in that mass extinction. And to me, it's sort of like an eraser and a reset button. And I think of it as part of the beginning of our origin story, because if it hadn't happened, you and I wouldn't be having this conversation. If it hadn't happened, mammals wouldn't be dominant on Earth. We wouldn't have primates, et cetera. And the world we see out there, which the groups that rebounded, birds from a very small number of ancestors that flourished into the types of birds, there was essentially a bird radiation, a frog radiation, a mammal radiation after this. Snakes, my little favorite group. Snakes are a small part of the story before that. These groups of snakes I'm talking about, a lot of their action has happened in the last 20 or 30 million years. The invasion of Australia, the invasion of North America, the radiation to all these types. That's pretty young as evolution goes. And then humanity. And I said, well, there's another big event, which is the Ice Age, a really peculiar, really odd geologic period. And I don't need to spend your time going into why the Ice Age kicked in, but let's just say there was some really interesting geology behind that. And without the Ice Age, you don't get the technological apes that have podcasts and talk over Zoom. So the origin story is to sort of think about the geological and biological events without which we would not be here, most of which was discovered in the last 50 or 60 years. Yeah, you couldn't have told this story to our grandparents. Didn't know it. In fact, couldn't have told them much about DNA either. Okay. So the explosion of scientific knowledge is massive. I think there's a very concrete narrative we can say about our kind, mammals, primates, humans. What were the contributing conditions to all that? And that's the kind of thing I think that whether you're, an artist or, a business owner, or a lawyer, this is the piece this is what biology has to offer you. And some other things too. But the part that I think you should have is some picture of the planet you're living on and how humans got here. And there's been lots of stories about humans told over the millennia. But the one that we can tell in the last 50, 60 years, including our ancestors out of Africa, remember that was only solidified really 1959. Mm-hmm. So it's a incredible achievement of humanity to understand how we got here. And it beats any other story that someone tried to tell me a long time ago when I was a little kid. And I think this is a big thing that evolutionary biology has to offer. So that's what I'm throwing out there.
All right. Well, Sean, Carroll, thank you for your time.
Thanks, Nick.
Hey everyone, I want to take a minute to tell you about a really cool health monitoring device I've been using for several weeks. It's called Lumen, and it's a handheld, pocket-sized device with a sleek design. It measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs. It improves your ability to burn fat, which decreases your hunger levels and makes your body less dependent on snacking, and it can increase your energy levels by helping you develop a high-functioning metabolism. I use this device in the morning, before bed, and after meals and workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I need to fast each day to promote fat burning. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. The easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MIND, M-I-N-D, in all capital letters to get $50 off your Lumen device today.